Welcome to the latest edition of the Mind Gut Conversation podcast, a place to learn about the latest ideas from thought leaders in the area of optimal health, food, the science of mind-body interactions, and the environment. Today, I have the great pleasure to speak to Colleen Cutcliffe, the CEO and co-founder of Pendulum Therapeutics, a leading microbiome solutions company based in San Francisco. Colleen has over 20 years of experience leading and managing biologic teams in academia, pharmaceuticals, and biotechnology. She received her PhD in biochemistry and molecular biology from Johns Hopkins University and her BA in biochemistry from Wesley College. Welcome to the show, Colleen. So I would like to start with this question, Colleen. How did Pendulum get into the business of live microbial therapies, and what's been the steps from the initial studies to the current products that Pendulum markets?
Yeah, it's been a longer road than I think we thought it was going to be. But when we first started Pendulum, it's me and my two co-founders. We're all three scientists, technical founders. So I'm a biochemist, Jim is a biostatistician, and John is a biophysicist. And we were working at a DNA sequencing instrument company at that time. And really what we saw was the emergence of, fantastic academic work on the microbiome, and understanding that this is a new science where there's opportunity to actually create new products in a world where probiotics and yogurts have been on the shelves for decades, but there hasn't really been an opportunity to bring new ingredients here. And so the idea was, can we start to create novel products in the space that confer a health benefit that is different from all the things that have been out there for these 50 plus years? And the reason it's taken us a while is because, of course, the microbiome is a new science. And so there are many tools even that didn't exist when we started the company that we had to create ourselves. And I think that what we're very, very happy about is that not just us, but many other microbiome companies that are out there are showing that there is going to be this next generation of products that are derived from microbiome science and looking at mechanism of action of the microbiome and its relationship with the host that are going to create new products that allow us to help people. And for us, we've started with our flagship product, Pendulum Glucose Control, which helps people with type 2 diabetes. And we've recently released a second product, Akkermansia, which is a strain that you cannot find anywhere else and is really important to gut health. And so we're starting to see the beginnings of the realization of this vision of creating these novel microbiome interventions.
I mean, obviously on the market right now, as you mentioned, I mean, there's hundreds of products and supplements and which are being promoted as the best and the largest quantity and the largest variety. And there's always sort of this claim, that they're scientifically proven, but that's really, I personally feel that 90% of these products do not meet these criteria. They often do not meet these criteria by the main or professional organization, ISAP, that a probiotic can only be called a microbe that, when ingested live, has a demonstrated health benefit on the host. Many of these, these probiotics that have been marketed, they have been shown to have benefits on the mouse gut or on isolated cells, but actually on the human host, that's a sort of a different challenge really that's been. And so does your product or your products, do they meet this definition of the ISAP organization?
Yeah, I feel like you're throwing me a softball with that question, but yes, when we developed these products, it's really about showing efficacy in humans and what it does to the human host. And then also not just that, but also looking at the underlying mechanism of action by which we think the product is behaving. And so, I would ask when all of us sit in this problem, every consumer goes into a store and just these shelves and shelves of probiotics, how do you recommend people start to understand the difference between a label that says it's scientifically proven and a product that actually is scientifically proven? You know, what do you look for that gets beyond that, you know, those marketing claims?
Well, so it's a very difficult question because it's probably the most commonly asked question whenever I give a talk: which probiotic should I take? And so my answer on that has generally been, if you're taking something that gives you a benefit, a sustained benefit, you should probably stay on it. It works for you. The evidence for controlled clinical trials in humans is much more sparse. It's really laudatory if a company that has a candidate probiotic, if they're willing to take the risk and invest the money to do a clinical trial. That to me is a criteria. So there's you know, a couple of new companies that that are doing this, and we all know these clinical trials are extremely expensive. The big risk, if you have a successful product on the market and you do that study and spend $200 million, you might shoot yourself in the foot because it may turn out that it doesn't meet the criteria. And so I think this is one of the main reasons why we haven't seen more of those clinical trials. But I'm encouraged, and I strongly endorse these companies. So, you know, Seed is one, Danone is one. We've done a study with Danone on a specific question, not on gut health in general. So to me, that makes companies stand out and products stand out that that I would endorse. You know, and I think, or I hope in 5 years from now, there's more. The outcomes from these big clinical trials will be positive, and we will be able to say, okay, now there's 2 products on the market or 3 products that really have a health benefit, either gut health in general or specifically like what Pendulum has done on metabolics, on type 2 diabetes. So I think we'll get to that point. Sadly, you know, you could almost say the train is already out of the station, because consumers are not going to wait 5 years before these studies come out. They will want to buy products now, you know, that somebody can convince them, or which now often happens, an influencer on social media can convince them, not the science, you know. So I think that's yeah, and certainly, you know, Pendulum stands out with that, with a study in humans with type 2 diabetes. I mean, I think the other question I had is, what's unique about your products that differentiates them from other products on the market? I think, you know, we have partly answered that question now. I mean, so you've gone beyond, Pendulum has gone beyond the mouse studies into a human trial. You also have a study going on with the Akkermansia product. From what I know, you use a fairly unique clinical trial design for that. Could you explain that, how you evaluate this?
Yeah, I think, well, so, you know, with Pendulum glucose control, we did the clinical trial in sort of a standard way that you do with type 2 diabetes, and all the standard measurements that are taken there. And as you know, getting that published in BMJ was a very big deal for a probiotic to make its way in there. But that was a relatively straightforward, you know, double-blinded, placebo-controlled, randomized trial. When it comes to GI, and especially a strain like Akkermansia or some of these other novel strains that are coming out, I think it's not entirely clear which things people are going to benefit from, unless you're going after a disease state like IBD or Crohn's disease. I think in that case, it's very clear, you know, what are the inflammatory markers that you're looking for and trying to measure. I think when we think about GI issues that people have that haven't been bucketed as IBD, it starts to become a much more heterogeneous population. And, you know, you have people that are suffering from diarrhea, constipation, bloating, pain, anxiety, depression. So it's a much more complicated system. And these individuals have all been bucketed into the same bucket, and they likely are not in the same bucket. And so how do we start to think about the right solutions for these individuals? And I think that's something we've really been racking our brains over. So rather than approach this in the way that all these other products have, which is to just run a standard clinical trial, which has a very high placebo effect, as you know, and then put this product out there and tell people like, oh, well, there's a clinical trial, so therefore it should work for you. What we're trying to do is to take a different approach to the problem, which is to say that we have multiple mechanisms of action. We have strains which produce propionate, strains which produce acetate, strains which produce butyrate. We have strains which are able to help upregulate GABA. We have strains which are going after the free radicals and things like that. So we have different pathways of the microbiome that we think are important. And for you as an individual, we don't know exactly what is the problem that you're suffering from, and we don't just want to throw the kitchen sink at you. And so how do we bring people into a program? And this is what we're getting ready to launch, which is to bring people into a program and to have them run their own trial essentially on themselves. So try a formulation. We will give you the symptomatic test, which actually, Dr. Meyer, you're one of the co-creators of, and then also microbiome test and nutrition coaching, and really try to understand how is this formulation working for you and how is it not working for you, so that we can figure out what's the next thing to add on, and to really take people through this cycle where they're getting data back on what's working for them symptomatically and their microbiome, and pairing that with these different formulations to find the right thing for them. Because the other thing about your microbiome is, as you know, over time, it changes. And so what works for you today, many people may have experienced that, oh, I tried this product and it really worked for me, but after a little while, it fades, its efficacy fades, it doesn't work anymore. And that's because the microbiome is constantly changing, not just through our diet and exercise, but through aging and stress and circadian rhythm and all sorts of other things that we don't have control over. So we want to continue to provide people with a thing that's going to help them as their microbiome evolves. We want to evolve with them. And you can only really do that through this sort of a testing platform with the person's particular issues in mind. That's the end goal. And so we're going to be launching this GI repair solution, which is actually trying to help people and be their partner in finding the right solution for them. And we also know that through this data collection effort, we will start to be able to take this complicated heterogeneous population of people and sub-segment and begin to understand, should everybody just be in the same bucket, or are there different segments that can be helped with different formulations?
Yeah, no, this is really interesting. And I think it's also so, we all know that how different everybody is in terms of the microbial strains. Once you go down to the strain level, there's only about 10% similarity between individuals. So you would think that, almost everybody needs to take their own separate mix of strains of a probiotic. But there seemed to be one mechanism. I always hate as a scientist to sort of come up with this sort of one fits all concept. This phenomenon of this increased permeability of our gut barrier. So I'm trying to avoid the name leaky gut, which is in some ways, somewhat misleading, very popular. But so it seems that a lot of chronic diseases, possibly all of them, share this abnormality, that their gut permeability, which is a very complicated mechanism, is somehow compromised and lets bacterial or microbial inhabitants of the gut interact with sensors of immune cells, which then triggers either this low-grade immune activation limited to the gut, or it spreads throughout the body, something called metabolic endotoxemia. So this, in my view, is something that seems to be shared by not only a lot of people, but by a lot of different diseases. So there is this possibility that if you had a way to successfully target this membrane barrier or membrane permeability, that this would actually work for a lot of people. And going into the literature of Akkermansia, you could come up with this idea that this is one of those organisms, that may potentially have a widespread application. But then there's things that at first glance, seem counterintuitive. One is, so Akkermansia, gram-negative bacteria that lives in the inner layer of the mucus layer, and lives from these complex carbohydrates or glycans that make up that mucus layer. So it's not the whole mucus layer, it's just one section of it. And so you could say, well, if they gobble down these sugar molecules, it's a thinning of the mucus layer, and it would actually have the opposite effect. It would make it leakier. So, I mean, we've talked about this before. I personally, came up with a couple of potential explanations, but being aware that in this science, rapidly moving science, this could change, in a couple of years or in a year from now. But the fact that they break down this inner mucus layer, what they do with these molecules, they make short chain fatty acids out of it, which are this, what I've called the aspirin of the gut, or of the body, which have these anti-inflammatory effects. And since they're being released right into the mucus layer, it could be that this is a very effective way of delivery. Even more effective than when you get this from digestion of fiber, which happens in the lumen of the gut. So that's sort of an interesting thing. And the other thing, that this concept that if they feed on these molecules, it would lead to a thinning, that's probably also not correct, because there's clearly a very powerful mechanism to compensate for that loss of certain fraction, which are these goblet cells in the colon that produce constantly mucus. And so it's what Akkermansia may do, it may just increase the turnover of that mucus layer and make it even potentially more effective. So yeah, I mean, I think, most consumers don't think about that, but for a scientist, when you read this first, they say it just doesn't make sense. So here you have a molecule that is actually seems to be bad for your gut permeability. And on top of that, it's one of those organisms that has increased with westernization. So, we've lost a lot of organisms, most likely due to the change in diet and other factors, but that one has increased in its abundance, probably as a compensatory mechanism. There's less fiber coming in through the diet. So in order for this organism to thrive, it has, it uses the body's own, molecules, carbohydrates, to break them down. So when you think the whole thing through, it does make a lot of sense, that this could be potentially a next generation probiotic with widespread applications, and may not need potentially the personalization that we need for other probiotics, which is likely. So I don't know what your thoughts about this. I mean, there have been other groups, scientific groups in Europe, that have studied Akkermansia and have contributed to our understanding of this organism. But yeah, as a scientist and clinician, I find this a really interesting topic.
Yeah, I do. I think it is interesting to think about Akkermansia as sort of a keystone strain. I mean, I remember, when I was in graduate school was when, P53 as a cancer, gene was starting to emerge, and was sort of unbelievable that you could have this one gene that, if it had mutation in it, seemed to be correlated with every kind of cancer out there. And I think what we learned over time is that you should not think about cancers as much about, where they happen to be located, but what their molecular composition is. And so you can start to understand, why did you develop a cancer? Because this gene was actually mutated, and it's in a keystone gene. So if you don't have the right P53, you all of a sudden start to have a high, susceptibility to developing cancer cells. And I think with Akkermansia, we're seeing a similar type of keystone concept, which is that because it plays such an important role in the mucin lining. I mean, think about, the protection of your gut as almost like the skin of your body. I mean, it used to be that if you got a cut and it got infected, that could be a reason why, people would die. You essentially could never get cut on your skin because all of a sudden you've got this permeability of things coming into your bloodstream. And I think when we think about the gut, it's very similar, which is that lining is so important because it really is the thing that keeps certain things inside and certain things outside. And so compromising that, you could imagine, compromises a wide variety of different mechanisms within the body. I think, so I think, first of all, Akkermansia is emerging as a keystone strain also because of its correlation to a variety of diseases, where you look at healthy people versus people with a bunch of different kinds of diseases, and the people who are sick appear to be lower in Akkermansia. So it looks like Akkermansia is playing a more prevalent role than just a singular disease. I think your point, which is that, hey, it doesn't feed on fiber, but rather it feeds on, the mucin layer, is also very interesting and produces these short chain fatty acids. So if you don't have enough fiber in your diet, if you're on, for example, an all meat diet or the keto diet and all these different things, you have the ability still to produce short chain fatty acids through Akkermansia. And then I think, this third thing, which is that turnover, the degradation of the mucin layer is likely a part of the mechanism by which you have a healthy gut lining. That is something that I think is super interesting. And I'll also give the caveat that you gave up front, which is that this is still a very early science. So everything we say today could be proven wrong in a couple of years. But, I did my PhD work at Johns Hopkins on the ubiquitin pathway. And the ubiquitin pathway is about turnover of proteins. How does your body get rid of proteins after a certain amount of time? And if you can't do that well, there are a bunch of different diseases, including death, that come out of that. And so thinking about, I was sort of saying, it's like your couch. If you're going to upholster your couch, you don't just put the new upholstery on top of the old one. First, you take the old upholstery off, then you put the new stuff on. And so being able to take the old stuff off is as important to having the right gut barrier at the end of the day as putting the new stuff on. And I think that we're going to learn a ton about Akkermansia on that front. And maybe the last thing I would say, so, production of short chain fatty acids, the degradation of the mucin at the right time. And then the third thing that's interesting about Akkermansia is that it does contain the gene for potentially increasing GABA production. And so this is, again, a very, very early stage, but, glutamate decarboxylase is the key enzyme to producing GABA. And we don't know why Akkermansia has the gene that encodes for that. And in what circumstance is it maybe contributing to the gut-brain relationship? And I know this is your field of expertise, but I think there's something very interesting there also when we think about the keystone or the ability for Akkermansia to play a role in a wide variety of different health issues.
Yeah, also, I mean, one thing that's interesting, and it's kind of unique about this organism because it lives in the mucus layer, so it's closer to the lining of the gut with all its sensors and receptors that's sticking out there. So, I mean, we don't know the last word on this GABA production by microbes yet, if they make it in sufficient concentrations to the brain, or if they act on vagal afferents or on cells in the gut lining that are innervated. But I mean, being in the mucus layer, it makes them closer, that production of GABA closer to mechanisms that can transmit that signal to the brain. So
absolutely. And just to add to that, just to build on that too, we found that the one of the primary mechanisms for pendulum glucose control is the production of butyrate. But in order for butyrate to work, it has to bind to the G protein coupled receptors that are sitting in that mucin layer. And we found in our clinical trial that if you didn't have Akkermansia in that formulation, you didn't see as high of an impact on blood glucose control. And so in order to really improve your blood glucose spikes, you needed to have the butyrate production paired with the Akkermansia. And I think it's because you need that localization right there at the mucin layer where the receptors are living in order to, I mean, it's a classic biochemistry problem, right? You have two things and you have to make sure they intersect with each other. And it's just a physical proximity that makes that possible.
One other question I had is in terms of like all the microbial organisms have multiple strains. I mean, we know from E. coli, it ranges from health promoting E. coli strains to deadly strains, enterotoxigenic, what's known about the different strains of Akkermansia and do these different strains have fundamentally different functions? Has that been studied?
Oh man, this is very, very early on. I think for a long time, people didn't even believe there were different strains of Akkermansia. They thought there's just this one and it has this particular role. And I think, more recently it started to become clear that, there are different types of strains. And I think, the definition of strain also comes into question. If you have, a single point mutation, does that make you a totally different strain? And obviously the way you would like to define strains is based on activity. So all these different so-called strains, if they're just different mutations, but functionally the strains do the same thing, do you really just bucket them as one strain that happens to have mutations? And I think those philosophical questions have not been answered yet, but certainly it's possible. And I think that's new science that's just emerging. And I would say that for us, this is really important because the strain that we use, the Akkermansia that we isolate and that we use in our clinical trial, it's very important as we manufacture it to make sure that it is still that same strain, because strains mutate over time. And so as we, grow our strains, it's an important part of our quality control to actually fully genomically sequence the DNA. That's not required for the selling of probiotics. You just need to have, in fact, PCR results showing that's the thing. But we really do the full genome sequencing specifically to get at this angle.
Mm-hmm. Let me come to the last question. We've touched on this a little bit, but I mean, how do you see the future of next generation probiotics? I mean, this is obviously a wide open field with some major players that have entered this, and likely we're going to see a lot of activity in that space. So, as from a pendulum standpoint, how do you see that field emerging in the next 5 to 10 years?
Well, I just think it's such an exciting time to be in this field because there are hundreds of new things coming out every day. And the question over the next 5 to 10 years is, how can all of us, companies, academics, clinicians, all work together to help educate the public so that they can understand what is the difference between a probiotic with real efficacy behind it and somebody who's just trying to sell me something to make a quick buck. And I believe that we are going to get there through education and products that actually deliver on a promise.
Yeah, thanks again, Colleen. This was a real pleasure talking to you. I should disclose at this point, I have been a member of the scientific advisory board of Pendulum. And, for me, I don't do this for every company that I'm asked by. And I'm kind of selective, and I think scientific evidence and rigor and transparency are definitely criteria that are essential for me before I spend my time doing that. And so I'm looking forward to, more studies in the future, more interactions, and, wish you good luck with this ongoing or starting trial on the Akkermansia study.
Thank you. And thank you so much for having me and for being an advisor to us. And it's been super helpful to get your guidance and, of course, the latest and greatest thinking on the microbiome and really starting to figure out how do we help create solutions for each individual out there and their needs.
Thanks, Colleen.